Okay, so let's talk about the liver. So here's your liver. We just saw it. We just went through all of the lobes and everything. Inside, internally, this is what it looks like. Okay, you've got these lobules, okay, liver lobules, if you will. They kind of look like hexagons. Each strand that you see kind of coming out of here is actually cells. If you look here, you can see the individual cells, and you can kind of see that they're making these walls, okay, out of these cells. The cells would be like the bricks in the wall. Each one of these is called a hepatic cord, okay? If you look in the middle, there's one central vein. And if you look at the corners, there's this kind of triplet going on here. I've got a red hepatic artery. Remember, hepatic artery is bringing fresh oxygenated blood to the cells. I've got the hepatic portal vein. This is the one bringing all of the stuff that I've absorbed to these cells, okay? And you'll notice right here that they fuse together to basically make one vessel called a hepatic sinusoid. Now, that vessel is going right by all of those cells that are making up the wall. The cells are going to pull anything that's important. Oxygen, it's going to pull, if I've got, like I said, nutrients that still need some processing, it'll pull those. It'll pull nutrients that they can use as their own fuel source as well. And if it's changed something to make it better, once it's done, it dumps it back into that vessel. Now, or sinusoid. The carbon dioxide, there we go, that the cells are making, that waste product, is also being put into that sinusoid to be carried. Now, see this? This is where that ultimately gets dumped, and eventually it goes to the inferior vena cava to get put back into general circulation. Now, the other, or the third member of this triad here is the hepatic duct. So bile is made in the liver. That is where it actually gets produced. But unlike the products that are going toward that center into the middle, the bile needs to go to either the small intestine or to get stored in the gallbladder. Those are my two destinations for that. So I don't need it to go into circulation at all. So the cells are also producing bile, but instead of just dumping it into this hepatic sinusoid, it's getting put into these bile canaliculi, into these little teeny tiny canals. And it actually, instead of going toward the center, goes out toward these corners where this hepatic duct harvests that bile. And eventually those merge together into the left and right hepatic ducts. Remember the two green ducts that I showed you, those two tubes, and gets put down into the hepatic duct where those two merge. Excuse me. So, the liver is actually held together by connective tissue, as well as the visceral peritoneum. Remember that layer. Microscopically, you've got these connective tissue septa, the sections that divide the liver into these hexagonal shaped lobules with the portal triads at the corner. If you look at every corner, there's a portal triad. You've got an artery, you've got a vein, and you've got the hepatic duct at every corner. The central vein of each lobule drains the hepatocytes into the hepatic vein, which drains into the inferior vena cava. Again, eventually this is going to make its way back to the heart and go into general circulation. The hepatocytes are the functional cells of the liver. Hepato, remember, means liver. Cytes means cells. So they're the functional cells of the liver that make up the hepatic cords, the walls that I just showed you over there. The sinusoids are the blood channels between those hepatocytes. You've got two types of cells. You've got the endothelial cells and you've got the Kupffer cells. The Kupffer cells are macrophages. They're immune cells, okay? Between the cells of the cords, like what you see here, I've got those bile canaliculi, okay? So ultimately, if I am getting taken to the liver, okay, I'm either coming in as nutrient-rich, oxygen-poor blood from the hepatic portal vein, or I'm coming from the heart as nutrient-rich, oxygen-rich blood from the heart. Both of these get mixed into that one big sinusoid that I showed you. Now, depending on what you are in the mix, if you're a nutrient, if you're oxygen, if you're a waste product, these are kind of the options. I'm either going to take you in and store you. Let's say that I'm iron that's been recycled. The liver takes it in and it stores it. It's going to be detoxified. Let's say that I eat something and it needs to be detoxified before I can send it. Liver does that. The liver can actually take some of these energy molecules and use them themselves as a way to keep the cell running. Or it can be used to make something new. So for example, let's say that I ate 2 Snickers bars. That's a lot of sugar, right? So my liver starts making glycogen. It starts taking those glucose molecules and making them into basically human starch, okay? So again, I can either get stored, I can get detoxified, I can get used immediately, or I can be made into something different. So, if I am released from the hepatic sinusoids, I go toward that central vein, to the hepatic vein, and eventually to the inferior vena cava, where I'm going to make it into general circulation. Let's say it's a nutrient, so it starts moving. However, if I am garbage that, nobody wants, or I'm bile that has been made, I get put into the bile canaliculi. It goes out toward the sides, not toward the center, to the hepatic triad, and I exit through that hepatic duct to be released in the bile. When we talked about red blood cell recycling, we talked about bilirubin, and I told you guys that that gets put into the bile so that it goes out as a waste product. So if you're good and you're useful, you get kind of sent to the center to go to the body. If you're either bile or you're garbage, you get sent to the bile duct to basically get either sent out with the poop, or if you're bile, to get sent out so that you can help with the emulsification of fats, which emulsify just means breakdown of fat. Okay, so we have 6 functions when it comes to the hepatocytes. So hepatocytes are liver cells. So if we're talking about the hepatocyte function, ultimately we're talking about the function of the liver, because what is your liver made out of? Hepatocytes. So it's the same thing. First thing, bile production. Your liver makes the bile. Period. End of story. It's what does that? So bile does a couple of things. It helps to neutralize and dilute the stomach acid. So remember, I told you that when things first go into that small intestine, your intestine isn't meant to deal with acid like that. So bile is actually a neutralizer. It helps to neutralize that. And it also kind of helps to dilute, dilute that out, so that the pancreatic enzymes can function. Bile salts also emulsify fats. They basically take big fat globs and make them into smaller fat globs. So, something else. Most proteins have a very narrow homeostatic window in which they work, meaning that if you're at the wrong pH, you're going to destroy them. The best way I can think to describe denaturing a protein: when you crack an egg, there's that clear part, and when you cook it, it turns white. That's because you're denaturing the protein. So when the pancreatic enzymes are getting released, if you were to just put them into that acid chyme that's coming out of the stomach, it would literally denature them. It would destroy them. They wouldn't work. So the bile actually helps to make that environment the proper pH for them to work in by neutralizing that acid. Okay, storage. It can remove glucose and store it as glycogen. I just talked about that. It can store fat. It can store different vitamins like A, B12, D, E, and K. It can also store copper and iron. So one of the reasons that if somebody is anemic, they tell them to eat liver is because it is a storage site for extra iron. So if you're eating liver, you're actually consuming iron. That's actually a good thing. Interconversion of nutrients. So it can actually convert some nutrients into other nutrients. So one of the big popular things, right, is Atkins or protein diet or whatever you want to call it. I hate to tell you, but your liver takes some of those amino acids and converts them into sugar, so that you have fuel so that your body can keep running. So when, you don't have any sugar, well, technically your body makes sugar. So that's the interconversion of nutrients. I can make one thing into something different. Okay, detoxification. Products of ingestion, products that you're putting into your body, or even sometimes products of metabolism, when things start breaking down, they can actually become toxic. Just in the processing of the molecule. So when it goes through the liver, through the hepatocytes in those hepatic cords, they can actually change the shape. They can add things to it. They can conjugate to something else, and it makes that thing less toxic. So it's detoxification. Perfect example, alcohol. You drink a beer, you drink whiskey, you drink a daiquiri, any of those things. You're drinking, all intents and purposes, poison. So you've got enzymes in your liver that actually take that and break it down. It's called alcohol dehydrogenase, and break it and make it into something that is not toxic. Now, can you overrun the system? Yes, it is completely feasible to overrun the system. If you've got more alcohol than you have enzymes to deal with it, then yeah, and that's how cirrhosis of the liver happens, because you're drinking so much alcohol, you don't have enough enzyme to break it down into something that isn't toxic. So the toxic chemical is actually killing the cells of your liver. Okay, phagocytosis. So remember those Kupffer cells? I said that they were immune cells, right? Sorry, there's a window over there and I keep seeing people pass by and I'm like, who's staring at me? Anyway, they're macrophages. So one of the things that they do is they eat. So worn out or dying red blood cells, white blood cells, bacteria, debris, the Kupffer cells are doing the same thing that macrophages do. They clean up. So technically, phagocytosis is a part of what your liver does because Kupffer cells are part of your liver. Synthesis of blood components. So it can produce new compounds that can be released into the general circulation. So we talked about blood proteins when we went through chapter 19. I talked about albumin, I talked about globulins, I talked about fibrinogen, I talked about heparin, I talked about the clotting factors. These are not made in your bone marrow, they're actually made in your liver, and then they get released into the liver, into the blood to function there. So it is responsible for producing those things. Now let's talk gallbladder. As I pointed out before, this green balloon looking thing here is the gallbladder. It has 3 tunics, layers. Tunics just means layers. You've got an inner mucosa that has rugae, the same type of wrinkly thing you got going on in your stomach so that it can expand and hold volume. You've got a middle muscularis that when you need bile out of the gallbladder, this squeezes so that it can empty. And then you've got the outer serosa, which is that same outer layer as everything in that abdominal cavity. So the cystic duct, remember duct is just tube. This is the tube that carries the bile from the gallbladder to the common bile duct. The liver is responsible for producing the bile. Hands down, that's a job for the liver. But my gallbladder actually stores bile and concentrates it 5 to 10 times more than what you originally get from the liver. It doesn't make bile. It receives it and it concentrates it and stores it. That's it. When cholecystokinin is released, it actually does stimulate the gallbladder to release bile. Okay. Now, for those of you that are going, so what happens when you remove the gallbladder? Well, your liver is still making bile, so you still have the capacity to digest lipids. That hasn't gone away, but that ability to concentrate that bile, so you need very little to actually do a lot of work, kind of goes away. So when you have something that is very, very high fat after having your gallbladder removed, you may not be able to make enough bile to actually digest it properly. So when, probably more information than you need, but when people who don't have a gallbladder eat very, very fat, high fat foods, let's say fried chicken with fried potatoes and all of that, even donuts, because they fry those, they tend to get diarrhea. And the reason they tend to get diarrhea is because you've got all of this fat content that wasn't absorbed and broken down still in the material that's getting turned into fecal material. So it becomes very runny and almost slick. I mean, imagine putting oil on something. It gets very, very slick. So they tend to have issues with eating very, very high fat food, because when they do, they end up with tummy trouble. Okay, so the pancreas. It's this little wormy dude right here, okay? We already kind of know this. We've talked about it previously. The pancreas has both endocrine and exocrine functions. Endocrine, because it's got the pancreatic islets that produce the insulin, the glucagon, and the somatostatin. Insulin and glucagon are responsible for controlling your glucose level in your blood. Somatostatin is basically responsible for controlling insulin and glucagon secretion. And it may inhibit growth hormone. We're not positive on that yet. The exocrine function, though, however, is what we're more interested in for our digestive system. So remember these flower petal cells that I talked about previously? These are the Acini cells. They are responsible for producing digestive enzymes. Okay, so they're making digestive enzymes. Those digestive enzymes are going to be involved, obviously, in digestion. So they make them and they basically dump them into this space here, and eventually it collects in these ducts. Again, remember, duct is just a fancy way of saying tube. So you've got the intercalated ducts, and they eventually merge to become intralobular ducts, and eventually they become interlobular ducts. I'm going to go back. And eventually they dump, I guess, into this pancreatic duct right here, okay, to be put into my small intestine, where it merges into the hepatopancreatic ampulla into the duodenum of the small intestine. Again, that's right at the beginning. That's the first part. So, the secretions coming from the pancreas are actually called pancreatic juice. Again, I know it sounds gross, but it's still what it's called. We've got two components to it, an aqueous component and an enzymatic component. The aqueous component, it's produced by the columnar epithelial cells of our smaller ducts in the pancreas. It contains sodium, potassium, and bicarbonate. Bicarbonate is an acid neutralizer. Okay, it's a buffer. It's going to help us to get that pH back into a more neutral range instead of being so acidic. Remember, I said that pepsinogen needs to be exposed to hydrochloric acid in order for it to work properly, to be converted into pepsin. When I change the pH using the aqueous component, pepsin stops working. It's not an acidic environment anymore. It just doesn't work anymore. But it does provide the proper environment for these enzymes to work. Okay? So these enzymatic components are produced by the acinar cells, the flower petals. They're important for digestion of every major class of organic food compound that we have. So when we talked organic chemistry in AP1, we talked about carbohydrates, lipids, proteins, and nucleic acids. Okay? All four of these have enzymes coming from the pancreas to help us to digest them. Okay? So, proteolytic enzymes. Proteo, protein, lytic, lysis, to break. It's secreted in an inactive form and must be activated when released to break down peptide chains. So remember we talked about unwinding the sweater and then taking scissors. This is another set of scissors. If we've got lengths this big, this is cutting it into even smaller lengths. Lengths, like with a TH, of amino acids. So something else, this kind of goes back to what we've been talking about this whole time. Remember that with enzymes, they don't know friend from foe. They don't know who's a good guy or who's a bad guy. So I don't want this to work until it's in the duodenum. I don't want it working in my cells or it'll start breaking down stuff in my cells. I don't want it working in the tubing or it'll start breaking down stuff in the tubes. So when I say it's inactive and has to be activated when released, that's what I mean. Pancreatic amylase. Remember, we mentioned amylase when we talked about salivary amylase. This is a starch enzyme. It breaks starch down. Going from a polysaccharide either into a disaccharide, pretty much into a disaccharide. Pancreatic lipase, lipid ACE, lipid enzyme. It breaks down lipids from into monoglycerides and free fatty acids, cholesterol, and other components. So I'm taking these big complex lipids and basically making them into smaller bits and pieces. And this is the only place in your entire digestive system where you can break down nucleic acids. Deoxyribonucleases break down DNA. Ribonucleases break down RNA into their component parts. There is no other place in your entire digestive tract that does that. Only your pancreas, okay? So, hormonal control. Secretin and cholecystokinin. Remember that they're getting released by that first part of our small intestine. Their release is stimulated by that acidic chyme entering the duodenum. That is the same thing that I said before. But it actually does stimulate the secretion of the aqueous solution from my pancreas. Now remember, that has the bicarbonate. It has my neutralizer in it. So that makes sense. Cholecystokinin also stimulates the gallbladder to secrete bile. And the cholecystokinin is going to get that pancreas to release its pancreatic juice that has all of the digestive enzymes in it. As far as neuronal stimulation, my neurons, my nervous system, just like with everything in our digestive system, it's the parasympathetic rest and digest, stimulating it, and sympathetic fight or flight inhibiting it working.